You're listening to The Sound of Science, Future Feasts, by the Weizmann Institute of Science.
I often find myself staring at a handful of soil and wondering, how much information is actually hidden in this dark, silent matter? To most of us, it's just dirt. But if you look closer, if you look through the eyes of a scientist, you realize that the ground beneath our feet is a living, breathing archive. It's a biological hard drive, storing a protocol that has been running for billions of years. Hi, I'm Professor Roee Ozeri, and this is The Sound of Science. Today we explore the invisible threads connecting the Earth to our identity. We tend to think of plants as standalone objects, green machines converting sunlight into calories. But a plant is not an island. It's part of a deep, ancient conversation with the world below. A dialogue of chemical signals and genetic trades that has been happening since the first roots touched the Earth. In the rush to feed a growing planet, we have accidentally interrupted this conversation. We've turned up the volume on production, but in the process, we've created a sort of biological noise that has made the plants forget their oldest friends. Together, we're going to trace the path of our food from the silence of the soil to the complexity of the human brain. We'll talk about how we broke a partnership that sustained life since the beginning of time, and how we're trying to fix it, not just by looking at the land, but by rethinking the very definition of a factory. We'll see how yeast can be taught to remember, and how our own immune system decides whether the world outside is a friend or a threat. Imagine the early 20th century. The world was on brink of starvation. Humanity was desperate for a solution. Then came the Haber-Bosch process, the industrial miracle that allowed us to pull nitrogen out of the air and turn it into fire for our crops. This fire allowed us to support billions of lives, but every fire leaves a mark. While we were busy saturating our fields with nitrogen, we did not realize that we were changing the fundamental rules of the microbial world.
One of the things that we found when we looked at microbes in the ocean and the soil is that their response to temperature is greatly affected by nutrient limitations.
Says Dr. David Zeevi, a computational biologist here at the Weizmann. David doesn't just look at the soil. He listens to the data hidden in the microbial communities. He realized that our footprint on the planet is much deeper and much more complex than the mere footprint of our shoes.
Now, humanity affects the world in many ways. One of the ways is biogeochemical flows, the amount of nitrogen and phosphorus, for example, we put in the environment. So ever since Fritz Haber and the Bosch company developed the Haber-Bosch process in the 1910s or 20s, we've been putting a lot of nitrogen into the environment. How much is a lot? Imagine all of the bacteria that fix nitrogen from the air and put it in the soil. We put as much nitrogen fixed into agricultural land every year, as much as everything else in the environment. And this has a huge effect on waterways and, of course, on microbes.
We've effectively doubled the world's nitrogen flow, bypassing a billion-year-old biological process. But David discovered that this intervention created a new kind of signal. These microbes are now acting like a biological crystal ball, operating months before a single seed is even planted.
Just by looking at the microbial communities in each of these microplots, we can predict how much wheat will grow at the end of the season.
David's lab found that these microbial communities are great predictors of environmental and agricultural health. And it's not just a few plots. It's the massive fingerprint we've left on nearly half of the Earth's land.
Agriculture is not just one field or two fields. About 10 to 20% of the land mass of Earth that's not covered by ice is covered by croplands. Another 20-30% are covered by pastures. So agriculture has a huge fingerprint on the land on Earth.
But here is the cost of our success. In our effort to make plants grow faster, we made them lonely. We gave them so many chemicals that they stopped recruiting their natural partners.
What my lab is trying to understand is how plants coordinate or regulate their environment in terms of bacteria, which bacteria will be sitting on or located or colonizing their roots.
Professor Asaph Aharoni looks at the plant as a conductor of a microscopic orchestra. He spent his career uncovering how plants use a complex chemical language to recruit the right microbial musicians, an orchestra we've accidentally muted.
They don't want every species to do it. Some of these bacteria are harmful to them. So they have their way, a few major ways to control which bacterial species will be colonizing their roots. And one of the major ways by exudation. So they secrete or exude out of their roots all kinds of chemicals that will actually attract the good bacteria and avoid the bad bacteria to attach to their root. So there's a certain community in surrounding the roots and sitting on the roots and even entering the plants.
Modern wheat, as David Zeevi found, has lost this conductor's baton. It grew tall on our fertilizers, but it forgot how to speak the ancient language of the soil.
Microbiome of modern wheat was markedly different from all other wheat accessions. So somewhere in modernization, wheat changed its ability to recruit certain microbes. And this could be fundamental. Having changed the microbiome of these plants means that we may have changed their ability to recruit more nutrients from the soil. Maybe when we evolved modern accessions of wheat, we lost some very basic properties of the microbiome that could help us in the future grow wheat more sustainably.
This loss of connection isn't just about efficiency. It's about the very survival of diversity. And it makes me wonder, if we rewind the tape of life, would we choose this path again?
Stephen Jay Gould proposed a thought experiment. What would have happened if we would replay the tape of life?
Dr. Shahar Rezenman explores this question through a famous thought experiment. Shahar is an evolutionary biologist who likes to think in centuries, fascinated by the idea that if we could press play again, the world might look entirely different.
He suggested, let's think of taking 500 million years back. Let's replay the tape of life any number of times and let's see if evolution will repeat itself. Gould actually suggested that it won't happen. He basically thought that the role of chance and randomness in evolution is really important and that every time that we will replay the tape, something else will develop.
Shahar's work in the lab shows that diversity is our only true insurance policy. But diversity needs time. When we force change too quickly, whether through intensive farming or climate shifts, the tape doesn't play replay. It breaks.
One of the most important parameters that we can check about population is the diversity in this niche. If the rate at which the temperature will rise will be fast, we will lose much more species, we will lose much of the diversity that exists on Earth. But if we'll do it more gradually, we will basically keep more diversity. And we will give more species a chance to adapt and survive those periods.
If the soil is struggling and the environment is changing too fast for traditional farming, maybe we need to change the venue. For 10,000 years, farming meant looking down at the ground. But what if we started looking at the very molecules in the air? Imagine a farm that doesn't need soil at all.
Microorganisms, tiny organisms that actually they can grow using carbon dioxide from the atmosphere as their source of carbon.
This is Sam Lovat, a PhD student in the lab of Professor Ron Milo. Sam is chasing a vision called solar food, capturing sunlight to split water, creating hydrogen, and feeding it to microbes that breathe in CO2 and breathe out high-quality protein.
And they can use an energy source like hydrogen. Now, the idea of using hydrogen to power the growth of these microorganisms sounds very interesting. It sounds a bit sci-fi, but it's possible. You could use solar powers. You could use electricity from solar panels to do this. And this is very efficient in terms of solar panels are very good at converting sunlight into electricity.
It's a radical act of conservation. In a world where water is becoming the most precious resource, this lab-based approach could change the global math.
Traditionally, 1 kilogram of, say, wheat or of soybeans uses a few hundred liters of water. It consumes a few hundred liters of water, which is quite substantial. With these kinds of new technologies, that footprint could be reduced by more than tenfold. It could be something like 10, 20, 30 liters of water per kilo. And that's really quite substantial.
But life in a lab has its own challenges. evolution doesn't find the perfect solution immediately.
What we found is that the first solution, the thing that the cells do immediately, is to duplicate a chromosome.
Dr. Orna Dahan, a staff scientist who has watched cells evolve for over 2 decades, discovered that when life faces a new stress, it often finds a quick fix first, a heavy genetic patch before it finds elegance.
However, if you let them continue to grow, they lose this extra chromosome and they replace this solution by more pinpoint solution. Duplicating a chromosome is a solution that can be found very quickly. However, it's not an ideal solution. It helps them to deal with the high temperature, but it also have a very, very large cost to the cells.
To find the most efficient procedures for a future food, Orna Dahan takes this evolutionary struggle to the extreme.
We are taking different natural isolates, yeast that were isolated from all over the world, mix all of them together in a test tube, so we have a big orgy going on in the test tube, and they can mate based on their preferences, if they have preference.
A yeast orgy, a massive genetic experiment to see which superhybrid can thrive in the high-stakes environment of a food tech factory. This is what Shahar Rezenman calls evolutionary intelligence. We aren't just coding machines. We are collaborating with the inherent intelligence of life to solve our most human problems.
I like to call it evolutionary intelligence and not artificial intelligence. We use the actual evolution, the selection mechanism and everything in order to find out the best solutions. We don't rely on any other information that existed in the past.
But our story doesn't end in a laboratory. Eventually, the food we create, whether it's air-based flour or engineered protein, enters the most guarded frontier of all, the human body. Our immune system is a sentinel, asking one question: is this a friend or is this a foe?
What we know about food is actually very interesting. We know that for decades now, that if you eat something, some antigen, some protein, you will actually be protected from initiating inflammatory response to the same food.
Dr. Ranit Kedmi is a systems immunologist who explains that oral tolerance is the silent permit that allows our body to accept the outside world as fuel. Without it, a simple act of eating would be a constant inflammatory war.
So basically, this phenomena is named oral tolerance. Your body will be restricted from inducing inflammatory response because it thinks it's food and we should tolerate food. And that is to ensure safe consumption of dietary antigens, because otherwise you won't be able to eat anything.
Ranit discovered a specific cell, the tolerocyte, that manages this delicate diplomacy. But once the immune system says yes, the brain takes over.
The dog salivates when you ring the bell because it's anticipating getting the food.
Dr. Yoav Livne from the Department of Brain Sciences tells us that our brain is a prediction machine. It doesn't wait to taste the meal. It starts preparing the body the moment we see a menu.
Pavlov got the Nobel Prize for discovering that the dog's stomach releases digestive juices, its pancreas releases insulin, and many other physiological changes happen in anticipation of feeding. And if these changes don't happen, the body doesn't deal well with the food. You're reading a menu, you know that this is food that can enter your body. And then the insula is preparing this basically command to the body that says food is coming, let's prepare, start salivating, start releasing gastric juices, start releasing insulin.
This brings us back to where we began, to the hidden protocols. Just as David Zeevi found a hard drive of information in the desert soil, Yoav finds one in our gut.
The gut microbiome is sort of another layer on top of that. So now you have microbes that also secrete molecules that can be sensed through the blood and all the way to the brain.
Our internal microbes are talking to our brain, influencing our mood, our satiety, and perhaps our own very identity. They're another layer of information in the ancient dialogue between our body and our world.
Basically, they're interacting with these pathways that are already there, and they're also affecting how food is metabolized. So basically, they're affecting everything in that way.
And just like the microbes in the dirt, our internal companions are part of an ancient race for survival, striving to be the fastest in an environment we are constantly changing.
Microbes always race against their compatriots. They are in a constant struggle to be the fastest in their own environment.
We started today in the dirt, and we ended in the brain. We've seen that food is more than just calories. It's a protocol of life that connects the deep history of the earth to our own very survival. We've learned that when we break the link with the soil, we lose more than just nutrients. We lose a piece of our resilience. But we've also seen that through evolutionary intelligence, solar technology, and a deeper understanding of our own internal guards, we are finding new ways to rewrite the script. We often make the mistake of thinking we're here alone, calling all the shots. But the truth is that we are part of a sophisticated and delicate fabric. We are surrounded by, and in deep symbiosis with, trillions on trillions of bacteria. This bacterial biomass, which far exceeds that of humanity, is everywhere, in the soil, water, and air that surround us, even in our bodies. Our relationship with these invisible allies will determine the future of food. Next time you look at a menu or walk through a field, remember, there is a hidden world working beneath the surface to keep you whole, and we're just beginning to learn its language. Thank you for joining us on Sound of Science. I'm Roee Ozeri. Goodbye.